In another area of our lab, we're very focused on addressing cancer. And the way that we do this is by designing nanoparticles which can deliver drugs directly to cancer cells. We have a number of tools in our toolbox to do this with. And some of our work involves designing polymers that wrap a drug in a very specific way. Another approach, however, is to go back to that layer-by-layer approach in which we use charge to wrap our drug onto a nanoparticle core. This is something that allows us to take an existing nanoparticle material that is already FDA approved, like a liposome or a vesicle. We can insert into that nanoparticle a chemotherapy drug that is normally going to be highly effective over a range of different cancers. Now, a number of the more aggressive cancer types are ones which have genetic modifications which allow them to survive even in the presence of those chemotherapy drugs. So what we're doing is designing a way to address that drug resistance in these really aggressive tumors. So we take that nanoparticle in which we've encapsulated the initial drug and we wrap around it layers which contain siRNA. siRNA will block the genes that allow these cancer cells to survive. Once we've wrapped that layer on, we have a need to protect that siRNA because it degrades very rapidly in the bloodstream. So we wrap it with a positively charged layer of polymer, and then on top of that we put a final layer of polymer that's negatively charged. It's actually a natural polysaccharide. It already exists in our body. It's called hyaluronic acid. And the magic of that final polymer is that it's very highly negatively charged, which means that it won't interact with a lot of the other cells that it encounters in the bloodstream. And it also has a lot of water around it. It creates a kind of cloud of water around that nanoparticle, which allows the nanoparticle to exist in our bloodstream without being eliminated by the body's immune defense system. But when it gets to the blood vessels in the tumor, there are tiny holes in those blood vessels because of the way the tumor has developed. And that allows the nanoparticle to get into the tumor. Because we've introduced molecules on the surface which also bind to cancer cells, we can get the cancer cell to take up that nanoparticle. Once we get that, we have the release of the siRNA inside the cancer cell. That lowers or eliminates the defense mechanism that those tumor cells have. Then the chemotherapy drug arrives, and its release leads to the death of those cancer cells. So we found this to be very encouraging. We've had encouraging results in our animal studies, and we're hoping to move this forward for more extensive study. In our lab at the Koch Institute, we have other simpler methods of wrapping drugs for some of the less aggressive tumors. But for these specific tumor types in which we really need a combination of the siRNA and the chemotherapy drug, the layer-by-layer approach is really powerful for us. There are several tumor types for which this kind of approach is relevant. Really aggressive tumors can be developed in triple-negative breast cancer, in particular because we don't have a very targeted approach for a triple-negative breast cancer at this time. We also see this to a large extent in advanced serous ovarian cancer. That is an advanced ovarian cancer type that tends to show up the first time. It gets eliminated by the first onslaught of chemotherapy drug, but it recurs in something like 70% of women. And in that recurrence, it has evolved into a genetic form that is resistant to the chemotherapy drug. So that is one of the areas we're particularly interested in addressing. We also see similar kinds of effects in non-small cell lung cancer. This is one of the more aggressive lung cancer types.